Good afternoon, everyone. Let's get started. This week, we're going to be learning about the immune system, and there are a lot of moving parts involved in the immune system. So hopefully, I can try to help you make sense of it all. As usual, if you have any questions, please do not hesitate to ask. First one, list 4 characteristics of innate immunity. So it's a good idea to have a quick refresher on what immunity is. So basically, you have your immune system as a whole, and that gives you the ability to defend the body against foreign pathogens. And your immune system is separated into what you are born with. So that's called the innate immune system. So innate immunity. And then there is going to be the one that you are going to have to acquire throughout life. And it kind of changes every time you get exposed to a pathogen. So this is called the acquired. Or because of its ability to change, to evolve over time, we also call it adaptive immunity. Okay, so the two branches actually have interactions with each other. They don't work in silos, they work together to give you a full protective immune system. So we're going to start with the innate immunity. We want to have 4 characteristics of innate immunity. So does anybody know what I'm referring to? Any answers? How would you characterize innate immunity? Barriers to entry would be an example of innate immunity. Yeah, but looking for characteristics, things that we can use to describe the nature of innate immunity. Maybe I'll do this one, and then, once you guys are all warmed up, we will come back to this. So, innate immunity is non-specific. It is not designed to fight a particular type of pathogen. It's more like a non-specific general type of defense that will work against a wide range of pathogens. Okay, they would work immediately. Okay, it is inherited, and it has no immune memory. It doesn't remember what you have fought off. So next time you encounter the same thing again, you will have to fight it off from scratch. And so these are the 4 characteristics of innate immunity. So we have various mechanisms. Basically, mechanism just means like different ways of defending the body. And here, the first one is barriers to entry. The best defense is just to keep the stuff out. Don't let them come in at all. Then we don't have to fight them. We don't have to deal with them. Just keep them out of the body. And that would be the purpose. I'll do purpose in blue, and then I will do description in red. Okay, so the purpose, just try to zoom in and fit everything in the box. The purpose of barriers to entry is exactly what it sounds like. Okay, prevents pathogens from entering the body. Okay, but in addition, we can also prevent them from attaching to the body. Okay, so don't let them in. Okay, and if they do come in, don't let them attach. Don't let them gain a foothold, so to speak. Okay, and so the description would be there are like different types of barriers, right? There are physical barriers, physical barriers. There are chemical barriers. Okay. Any examples of physical barrier? Yep. We have skin, intact skin. Skin is no good if there are breaks in it. Wherever skin is not possible, like your eyelid, like your eyes, surface of the eyes, for example, we would have mucous membrane instead. Okay. Ciliated cells, yeah, that's another good one. And then there's one that people usually forget about, and that's the normal flora. Okay, normal flora are these good microorganisms that lives on us and in us, and they take up so much space that there will be no additional space available for the bad ones to attach to. Okay, so those are your physical barriers. And in terms of chemical barriers, we have lysozyme, which is a type of enzymes that kills bacteria. Lysozymes in tears, sweat, right, perspiration, and saliva. Okay. We also have hydrochloric acid, for example, in stomach. Okay. So these things should keep the bad guys out, but of course they're not perfect. If they were perfect, we would never get sick. But as you know, inadvertently, we do get sick every now and then. So when they do come in and establish a foothold, we have other mechanisms to try to get rid of them because before they cause too much damage. And first one here is inflammation. Okay, so the purpose of inflammation is to recruit more white blood cells and plasma protein to the site of infection. Okay, so you want to get more white cells there to fight off the invading pathogens. You want to get more plasma proteins there to either help with the repairing or with fighting off the infection. So example of white cells that will be recruited include neutrophils, right? These are macrophages. These are phagocytes, I mean, that would be able to eat up bacteria. We can get some macrophage. Okay, so both of these are going to be important for eating up the pathogens. And then examples of plasma proteins that's going to help would be things like complement proteins. If you don't know what that is, I will explain in a bit. We probably going to recruit a little bit of clotting factors, maybe, if you have a cut, right? Stuff like that. And the description would be inflammation is triggered by the release of what's the chemical that's being released to trigger inflammation? Yeah, Tyler's a histamine. That's correct. Inflammation is triggered by the release of histamine by mast cells. And mast cells are kind of related to basophil. Basophils are going around in circulation, but mast cells are more like fixed in tissue. Okay, so this would result in, number one, vasodilation. That means the vessel is going to get bigger. And number two, we're going to have increased permeability of the vessel. Okay, so they're more leaky, and then they're going to be bigger. Right? So it helps to increase blood flow, which would then carry more white cells and plasma proteins to the site of infection. So hopefully you can fight up the stuff that made it inside before it causes too much damage. Okay. So histamine is also one of the things that cause you to have allergies, right? So it's allergy seasons. My nose has been nonstop running for a couple of weeks now, and the drugs that I take for the allergies, they are antihistamine, right? That would subside some of these symptoms. But antihistamine also makes you sleepy, unfortunately. Okay, fever. Fever is the purpose of fever is to increase body temperature, right? So normal body temperature is 37 degrees Celsius. And then if you have anything above 38, that's a fever. So how do you trigger fever? There are pyrogens such as prostaglandin. Will act on the hypothalamus in the brain to increase body temperature. Okay, so pyrogens are anything that would trigger a fever. There are different types of pyrogens. One example is prostaglandin, and it will go to the part of the brain called the hypothalamus, which you will learn more about later on in the brain unit. But hypothalamus is the master gland of the body. It regulates many things, and one of the things that it does regulate is going to be the body temperature. So the prostaglandin will tell the hypothalamus to turn up the temperature a little bit, and by increasing the body temperature, it helps to fight the infection. Okay. Next, we have non-specific phagocytes. Okay, so these are just phagocytes that are going to be walking around and mopping things up. Things are not supposed to be there. The purpose is to engulf. Oh my goodness, this is way too thick. Engulf any foreign. I should say engulf any cells that has foreign antigens on them. Okay, remember we have our own antigens, and then foreign cells, they have antigens. If their antigen does not match what we have in the body, the white cells will basically attack them and eat them. So the white cells, they tend to recognize what's called PAMs. Okay, so PAMs are pathogen associated molecular patterns. Okay, so these are a set of molecules that the white cells would immediately pick up and know that they are dangerous, and then they will eat up the cell. Okay, and then another function of non-specific phagocytes is that some of the phagocytes will help link innate immunity with adaptive immunity. Okay, through what we call antigen presentation. Okay, so let me briefly explain this after you finish copying it. I'll give you like a moment or two. All right. So there we have a phagocyte. Okay. And we will use macrophage as an example. Okay, because neutrophils cannot do this. Okay, macrophage can do that. And here, you know, we have a macrophage here, and then we're gonna have some bacteria here. Okay. So because it is a phagocyte, it will go ahead and eat them up. Okay, so afterwards, what's gonna happen is the phagocytes will eat the bacteria. Okay, they will surround them in little vesicles. Okay, let's just say it eats two. Okay, so this process, it is, as you know, called phagocytosis. Phagocytosis. Okay, it's gonna eat them up. And why would they eat them up? Well, because on the surface of these bacteria, there are probably some PAMs, right? Some pathogen-associated molecular patterns. Okay. So afterwards, what will happen is the phagocytes, the macrophage rather, it's going to do what's called antigen processing. And of course, all this is explained clearly in the lecture recordings. So antigen processing is going to allow the macrophage to break down the bacteria and display some of the bacterial antigen on its surface. Okay, so these are bacterial antigen. Like some of those PAMs maybe that we saw earlier. Okay, and then it is going to show these bacterial antigens to some of your T cells. Okay, so this process is called antigen presentation. Okay, and by showing this to the T cells, you will be able to activate them and basically trigger your adaptive immunity. Okay, so not all cells are able to do what I just described. Phagocytes is a type of cell that could do this. And for cells that are able to do this thing, we call them antigen presenting cell, okay, or APC for short. And so macrophage is an example of antigen presenting cell. Any questions so far about any of this? I know it might be a little bit confusing, but please ask me something if you have any questions.
I'm going to use my mic for this one.
Yes, please.
Because the macrophages are presenting the bacterial antigens, what's preventing the rest of the body from then attacking those macrophages?
That is an excellent question. So let me just reiterate what Tyler's asking. So earlier I said, right, where did I say this? I said the phagocytes will engulf any cells that has foreign antigens, right? So the bacteria has foreign antigens, and so the macrophage knows how to eat them. But once the macrophage puts the bacterial antigens on them, how come another macrophage is not going to go attack them, right? That's basically what the question is. Remember, this is an oversimplification of the whole process because we're trying to do an introductory course. In reality, these little things are actually placed on top of special receptors. Okay, they're called like MHC receptors, and so when the bacterial antigen is placed on these special receptors, then the cells know that this is part of our team. Okay, we should not be attacking them, right? So by putting it on one of these special markers are actually part of the body antigens. And in doing so, it distinguishes itself from when it is just, on the bacterial surface alone. Does that make any sense?
Yes, it does. Thank you.
Okay, great. All right, moving on. We have another cell here, which is the NK cell. Okay, what does the NK cell stand for? It has an awesome name. It is natural killer cell. And the function of the NK cells, the purpose of the NK cell is basically to kill infected cells. Okay, so sometimes if a cell is infected by a virus, and just a little bit of review of what a virus is. Let's say this is your cell, and then when a virus comes, okay, it will inject its genetic material into the cell, and then the cell is going to make little viruses and put them together. Okay, and then ultimately the cell is going to burst open, and all these baby viruses are going to come out. Okay, so when a cell is infected with a virus like that, they usually put on some distress signal on the surface. They're like, okay, like, please help me. I'm actually infected by this virus. Please kill me before I turn into a virus factory. Right? And so that's what the NK cell will do. It will come and then basically kill it off before it, hundreds and hundreds of thousands of viruses are going to burst out from the cell. It's like, zombie movie, right? If your friends got bitten, right, before they turn, they would ask you to maybe, put them out of the misery so they don't actually turn into a zombie. It's kind of, the same kind of concept here. In addition to being able to kill infected cells, they are also going to be able to kill cancerous cells. Okay, so and cancerous cells. Okay, but this is very, very limited ability. Right, one of the reasons why it is so hard for the immune system to fight the cancer cells is because the cancer cells is something that originates from within your body, right? So a lot of the surface antigens, actually, I would say most of the surface antigens are going to be same, if not very similar to all your other cells. So they would fail to trigger your immune system. Okay, but there are some subtle changes, and sometimes the NK cell might be able to pick them up and destroy them. But as you know, right, many people still get cancers. So this is definitely not a perfect system. Okay, so how does it work? Well, NK cells will trigger apoptosis, okay, which is programmed cell death in target cell. Okay, they would touch them, and they would basically create a cascade of chemical signals, which ultimately trigger cell death in these cells. Okay. Last but not least, we have our complement proteins, and we mentioned about complement proteins earlier. When you want to recruit the plasmin protein, right, it includes the complement proteins. So the purpose of the complement protein is to enhance. I think the notes use the word synergize, kind of mean the same thing. Enhance other immune responses. Okay, and how it works is basically it's a series of chemical reactions that relies on turning one on, which in turn triggering the second one, turning the second one on, and then turning the third on, so on and so forth. And there are many complicated pathways and outcome. But in the lecture, I gave you 3 examples, right? So for example, the complement proteins can recruit more WBC, right? So it's going to leave a chemical trail for the white cells to follow, so they know exactly where to go. Okay, so let's say the white cells just came out fresh from the bone marrow, and how does it know to go to your your thumb, okay, where you had a cut, right? Bone marrow is a long way from the thumb. And so, the complement protein is going to leave a chemical trail, right? Like these breadcrumbs for them to follow to get to the site quicker. And other function is they are able to tag pathogens to enhance phagocytosis. Okay, so by tagging the pathogens, it highlights them and makes it more visible to the white cells, and they're going to be able to find them quicker and eat them quicker. And they also can form these membrane attack complexes, okay, or MACS, which would then punch holes in bacteria, to punch holes in bacteria. Bacterial cell wall. Okay. So there you have it, the 5 mechanisms of innate immunity. Okay, let's do some polls. Let's do some simple stuff here. Where's my poll button? There we go. By the way, the first quiz was pretty well done. Class average was quite high, pretty satisfactory. Hopefully you guys will continue to do well. Okay. Most of you got it right. It is the ciliated cells, right? The stomach acid, the lysozyme, and the tears, they are all examples of chemical barriers. This is another one. Okay, anyone else? Okay, the correct answer is localized swelling. Okay, WBC enters from tissue into bloodstream. Actually, it's the other way around, right? You want the white cells to leave the bloodstream and go into the tissue, right? That's where the bacteria is going to be, right? So the correct answer is A. Okay, next one. PAMs are found on where do we find PAMs? Right, PAMs are found on pathogens. Okay, these are pathogen-associated molecular patterns, as the name implies. they are found on pathogens. The phagocytes are the ones that will recognize the PAMs and go attack them. Okay, so correct answer is pathogens. Okay, another one here. Yep, they are going to be formed by the complement proteins. We literally just wrote them down, right? Form membrane attack complexes to punch holes in bacterial cell wall. Let's just do one last one before we go back to our study guide. In case you don't know what malignant tumor means, that means cancer, okay? Opposite of malignant tumor is benign tumor. Very good. Okay, almost all of you got it right. The NK cells. Okay, so again, if you look at the thing here, the NK cell can kill cancerous cells. So if someone has very low count of NK cells, they might be more prone to getting cancer. Okay, great. Thanks for the participation. Moving on. List 3 examples of physical barriers. We kind of did that already, right? But you know, what the heck? Let's just write it down again. We talk about intact skin. I talk about mucous membrane. We talk about normal flora. Right? So like when you take antibiotics, right, you might kill off some of your normal flora. And as a result, you know, one of the side effects of antibiotics is you might get a little bit of upset stomach, right? A little bit of diarrhea, because some of the opportunistic bacteria now has more room to attach in your bowel, for example, and cause problem. They too will get killed off by the antibiotics, but you know, some might be able to escape because there's just so much of them, right? And cause you to have a little bit of an upset stomach. Not to worry, every time you kill off the normal flora, they will come back eventually, right? Sometimes you can take probiotics, right? You can either eat them in probiotic yogurts, or you can actually buy these drops from the pharmacy. They will say each drop contains, you know, 1.2 billion probiotics bacteria or something, and then you just take a drop a day, and it will give you healthy normal flora. It's not just in your gut, right? You have normal flora living on you as well, right? And having those normal floras prevents things like fungal infection. And again, it's just by taking up the space so that there are no additional space available for other things to grow on. Examples of chemical barriers, we talk about this as well. HCl, right? In the stomach to have low pH. There's the lysozyme, right? Okay, those are key examples of chemical barriers. List 4 characteristics of inflammation. If you cut your finger, it's going to be red, so redness. It's going to feel warm, right? It's going to be painful, and it's very characteristic to inflammation. It's that, you know, throbbing pain. If you ever cut your finger, you will know. At night when you're trying to sleep, like that finger is just like, you know, pulsating, right? That kind of throbbing pain is very characteristics of inflammation. And the last one is swelling. swelling. Okay. So how come we have these characteristics in inflammation? Well, that's primarily due to the effects of histamine on blood vessels. Okay, so earlier we mentioned one of the effects of histamine is that it will have vasodilation. So vasodilation will increase blood flow. Okay, by increasing the blood flow, it's going to result in redness and warmth. Right? So that's basically the consequences of this. Okay, blood is warm, obviously, and it's red. And it's like, you know, when you're embarrassed, your face flush, right? That's like the blood going to your face, right? So similarly, when the blood goes to where you have a cut, that's going to give you the redness. Secondly, it's going to increase permeability of the vessel. Okay, so we mentioned this is going to make it more leaky, right? More leaky. And it's a good thing in this case to make them more leaky because it will allow the WBC and plasma protein to exit the bloodstream easier, right? No point to bring them all to the location and they cannot squeeze out from the blood vessel, right? So you must also increase the permeability. So this increase in leakiness will also cause fluid to leak from the vessel, causing it to swell up, and all that excess fluid is going to be pressing on those nerve endings you have in the tissue, resulting in pain. All right? So that is that. Anybody have any question about innate immunity before we move on to adaptive immunity? Oops, sorry. Very good. Okay. Next, list 4 types of T cells, T cell effectors, and state their main function. All right, so let's just backtrack a little bit, okay? Just to fill you in case you need a refresher or you haven't seen the recordings yet. Let's just try to fill you in. All right, so remember that guy, the APC that we were talking about, the antigen presenting cell. And we've only learned one type of antigen presenting cell so far, right? The macrophage. So it ate up a bacteria, right? Super proud of what it did. So it's gonna put it on its surface. It's gonna show everybody, right? Okay, it's like my daughter. She make this origami boat, super excited. Got to show everybody that she sees. Okay, look, I made a boat. Okay, so this APC just ate a bacteria. It's all pumped up. It's going to show all the cells that it encounters. Particularly, it is going to show this thing called a naive T cell. Naive T cell. Okay, so what is a naive T cell? Well, this naive T cell is a T cell that has not been activated yet. Okay, so it came out from the bone marrow. It went to the thymus. It's going to stay there, but it has not been activated yet. It has not seen the danger of this world because they haven't been exposed to a pathogen yet. Okay, so that's why they're called naive like a kid. Right. So there are different types of naive T cells. What I mean, they're the same naive T cells, but what distinguishes them is the kind of receptors they have on them. Okay, so I'm just going to use colors to distinguish them. Right, so some have blue receptors, some have pink receptors, and I know I'm repeating myself because I already talked about this in the recordings, but I do apologize if you have to hear it again. But I do like to repeat myself. So these are naive T cells with different receptors. Now, these receptors are created through what we call gene shuffling. Okay, gene shuffling. Remember last semester when you were with me? If you were with me, and if you were not, that's okay as well. You probably have learned about genes, after you transcribe them, there are like, exons, right? So exon, and then what's between the exons? You guys remember?
Introns?
Yeah, the introns, right? So intron number 1, intron number 2. And then what I said at the time was, you remove the introns and then you join the exons together, right? Okay. But, that's not always the case. In the case of creating these receptors, okay, these are receptors. They're proteins, by the way. Receptors. Okay, the receptors are generated through gene shuffling. So just to keep it very simple, let's say I have 3 exons. Okay, so how many different receptors can I potentially produce? Anybody want to take a guess? I have 3 receptors. I have 3 exons in a gene. How many receptors can we produce? How many different types of receptors can we produce? Just throw out some numbers if you have any. Do you guys understand my question? Okay, Georgia said 6 receptors. Okay, anyone else? Three receptors. Okay, 4 receptors. Okay. And this could be a multiple choice question, right? You guys are creating all the different answers. Very good. Okay, thanks for participating. The correct answer is, well, let's find out. Okay, so the most obvious receptors that we can make is, we can take exon 1 and 2 and 3 and put them together, right? So exon 1 plus 2 plus 3, that's going to be 1 receptor there. Okay, but the body actually don't have to use all 3 of them. Maybe they just want to use exon number 1 and exon number 2. That's the second receptor. Maybe they want to use exon number 1 and exon number 3. Maybe they want to use 2 and 3. Maybe they want to use what else can they use? What did I miss? Help me out here. 1 and 2, 1 and 3, 2 and 3. Or maybe they just want to use 1. Maybe they just want to use 2. Maybe they just want to use 3. You see? So by choosing different types of combinations, you're shuffling the genes and therefore creating subtle yet important variations in the receptors. Okay, so how many different receptors did I create? 1, 2, 3, 4, 5, 6, 7 different types, right? Okay, so just by having 3 exons, you can create 7 different types of receptors, potentially, okay? But in reality, the genes that are reserved for encoding for these receptors, they have hundreds and thousands of exons, and therefore you can have virtually billions of different combinations when it comes to creating a receptor. Does that make any sense? Okay, so what is the purpose of these receptors, right? These receptors are going to have a key and log relationship with these antigens. Okay, so here I use color to represent it. It's just easier visually to see, but you can think of this, like if I zoom into here, you can think of the blue one as like having a triangle indentation, and then the pink one might have like a half of a hexagon, and then this one might have like, I don't know, I'm just making this stuff up, and then for the red one is going to have like a round, right? So it's actually shape that will be complementary to these foreign antigens, right? And of course, this semicircle won't fit here, so that's not the right cell. It won't fit here, that's not the right cell. And remember, the APC is just going to constantly presenting these foreign antigens to the naive T cells until it is able to find one that has the correct receptor. So this one is like a semicircle, so this would fit nicely in there, and that would activate a naive T cell. Does that make sense? Any question about any of the stuff that I just said? Please ask. Again, it's very hard for me to read the room because I can't physically see everybody's face. Okay. So once it has been chosen, right, this is the chosen one, this is the one, it is going to undergo clonal expansion. Clonal expansion. Okay, which basically means it is going to make many copies of itself. Okay, it's going to have many copies of itself. Okay, all of them is going to have the same receptor, right? Why does it do that? Well, because we found the T cell that has the capability of recognizing the invading pathogen, right? We don't want to make the ones that has the orange receptor. That's not going to help with the infection, right? We need the one with the right receptor. So we end up having a bunch of these naive T cells. And then they are going to differentiate. Right? I talked about this word last time. Differentiate is to transform, and it's a one-way transformation. It's to specialize into something, right? And that's what this question is about. That's how we end up talking about this whole mess, is because of this chart. We have to list 4 types of effector T cells. So these T cells, after these naive T cells, after they clone themselves, they can differentiate into 4 different types of effector cells. Okay. Does anybody remember what they are? Anybody got that far in the lecture? Yes, we have the killer T cell, not to be confused with NK cells, okay? NK cells is the stuff that kills infected cells and cancerous cells. This is killer T cell, and also sometimes it's called cytotoxic T cell. Same thing, okay? If killer T cell confuses you, you can use cytotoxic T cells. Is it called the T cells in the Resident Evil? I thought it was called the T virus, right? Or do they? I don't know. I don't remember. There's too many lores in that universe. But anyways, good reference there. The function of the T cells, the cytotoxic T cell, is to kill infected cells again. Okay. So, it overlaps a little bit with the NK cells, but this one is a little bit more specific. Here it said there is the helper T cell. Yes, helper T cell. And what does the helper T cell do? The helper T cell is going to activate naive B cells. Okay. We have the suppressor T cell. Good. Okay. What does the suppressor T cell do? Is it important to have the suppressor T cells?
To stop immune response when the infection is clear.
Exactly, right? So we need to stop the immune response when infection is cleared. Otherwise, they will keep on cloning themselves, right? We don't want, where was this thing? We don't want this clonal expansion to keep on happening, right? It's going to lead to uncontrolled cell division when the infection is over. So that's why we need this suppressor T cell. It's gonna stop the whole thing from continuing. Okay, and more importantly, perhaps the most important aspect is it will create memory T cell. Okay, and the memory T cell is going to stay in the body in case of future infection by the same pathogen. Okay, so next time you get infected by the same thing, you don't have to go through the whole presentation process to find the T cell. You already know what T cells are going to be needed to fight it off, and it could cause the duration of your infection to be much shorter. Okay, And creation of memory T cells is what creates lifelong protection against some of the diseases. So, you only get chickenpox once, for example. Then I know someone's going to ask, how come it's possible to catch COVID more than once, right? Don't we have the memory T cells? Well, there are all these variants, right? So like it's possible you get infected by one form and then, you can get infected by the Delta variant, for example. So it's not, a permanent solution for a whole class of virus, but for the individual strain that you got infected with. Okay. So those are the 4 types of cells. And just to reiterate, because there's so many things going on here. All these cells, right? All these cells are as a result of activating a naive T cell, right? So there's a naive T cell. I think it's French, right? So sometimes they have like this double I or something, but it doesn't really matter. So the naive T cell is going to become these cells. And in order to activate a naive T cell, what kind of cells do we need? What is the trigger for activating naive T cell? Anybody remember? Yep, it would be the APC. That's correct. The antigen presenting cell, right? The macrophage that we talked about. Okay. So let's continue with the story. List 2 types of B cells here. Sorry about the mistake. I never remember to fix them and post them. I should really get to work on that. So we have the naive B cell. Let's use hot pink now. Naive B cells. And we're going to activate them. They're going to go through clonal expansion just like before. But this time, instead of APC, relying APC to activate them, we're going to need to have the helper T cell. Helper T cell. From before, the helper T cell will activate the naive B cell, and they could turn into, as Adriana said, memory B cell. That's correct. Okay. So blah, 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 same as before. Okay. But more importantly, you're going to create these plasma cells. And plasma cells, they are responsible for mass producing antibodies. Okay. So just a quick work on how does antibody work? Okay, we talk about this in class. First of all, you should know that the antibodies are proteins, right? They have a variable part at the end. They're Y-shaped protein, Y-shaped proteins. They have a variable region at the top, and then there is a constant region. Okay. The analogy I use in class is that they're like a screwdriver, right? So the handles are the same for all the screwdriver, but the tips are going to be different. And so the variable region is the part that's going to be recognizing the pathogens. Okay. So how does the antibody work? Number one of the things it could do is it could form an immune complex. Okay, so which cause things to clump together, make them really big, and it's easier for phagocytosis to occur. They can tag the target, again, making it easier to be identified by phagocytes. They can neutralize toxins and viruses. Okay. And I think there's one more in the notes. They can activate complement proteins. Now, so basically that's what happens, right, with the entire immune system. Now, this whole thing about, you know, eating the bacteria, breaking it down, putting it on the surface, finding the naive T cells, so on and so forth, which ultimately leads to the production of antibodies, right? That's exactly what happens during the Rh sensitization that we talked about. Okay, except in the case of Rh sensitization, it's not bacteria that the macrophage is breaking down. It is the Rh positive cells, right? Okay, so these Rh positive cells might enter the mother's bloodstream, and then eventually they end up in the spleen or the liver. And in those places, there will be resident macrophage that will eat them up. And then it will go through this entire process, which eventually cause you to create anti-Rh antibodies, right? Okay, so this is going to take a week or 2 weeks sometimes. It really depends on whether or not you have the naive T cell present at the beginning. Okay, so remember, this is kind of like rolling the dice, right? You have, like I said, thousands and thousands of genes that you're shuffling, and you know, just by chance, you might not currently have a receptor that's available for whatever is infecting you, right? You will have to wait for that naive T cells to be made, right? You have a pool of them that exists within your body at any given time, but you might or might not have the one that has the correct receptor. Okay, let's say I get infected by like green bacteria. I don't have the green receptor at this moment, and I have to wait for it to be made, right? So during that time, you're trying to mount an immune response. The virus or the bacteria that you're infected with is causing harm in the body, right? So the whole question becomes, are you able to create the antibodies to activate the naive T cells before it has done too much damage? Okay, so in some cases, the disease is very, very virulent. It causes you to become very, very sick, and you simply don't have time to make the antibodies for that. And that's when you need to, you know, have medication that would, you know, kind of aid you in the healing process. I hope that makes sense. Okay, let's continue. List 4 characteristics of adaptive immunity. So unlike the innate immunity, adaptive immunity is specific. Okay, they are tailored to attack a specific type of pathogens. Okay, they, you know, if you get chickenpox, you're gonna get like anti-chickenpox antibodies. If you get malaria, you're gonna get anti-malarial antibodies. They are tailored solution for fighting a particular infection. Because they are tailored, because you got to make those antibodies, because you have to activate the naive T cell, it is not immediate. It's going to take some time, okay? And hopefully we won't get too sick while we wait for the antibodies to be made, okay? Another characteristic is diversity, okay? Because of the gene shuffling that I was explaining to you, you can virtually create, you know, billions and billions different types of receptors, which gives you the potential to recognize billions and billions of different types of antigens. Okay, I'm not saying that you have all billions of those in your body right now, but you have the potential to make them. Okay, and sometimes, right, some people would make a receptor that recognize something that is seemingly harmless. Okay, like pollen. Right, so you know, people who have allergies, right? What's basically happening is the pollens enter my body, and then I somehow have a T cell there that is telling me this thing is dangerous, and then we got all these, you know, inflammatory signals going on, giving me the itchy eyes, the runny nose, and all that stuff, right? And sometimes it's more deadly in some individuals. Okay, they have receptors that would recognize like certain antigens in peanut, and they have a nut allergies. Okay, and that creates anaphylactic shock, right? Very severe systemic inflammation. Okay, so sometimes it's the wrong kind of reaction. And finally, it has immune memory. Immune memory. Okay. List 3 effects of fever that will enhance immune defense. What's the point of cranking up the temperature? Well, it could increase metabolism. Okay, so by increasing metabolism, basically you increase the speed of chemical reaction, right? So many of the stuff that are going on in your immune system relies on chemical reaction. Case in point, the complement proteins. They are a cascade of chemical reactions. So if your body temperature is a little bit higher, then you can, get those reactions going a little bit faster and maybe form those chemical trails faster, form those membrane attack complexes faster. Another effect is it will reduce pathogen activity. Most pathogens are not good at functioning too well under high temperature. So that extra degree or two will slow them down, perhaps just enough for your white cells to have a slight advantage. And finally, by increasing your body temperature, you can maybe increase wound healing as well. Okay. So that is that. Any questions? Okay, that is wonderful. Why don't we take a break, and when we return, we'll do a little bit more. See you in a bit. Hello, let's keep going. I forgot to mention something. Over here, let's go back for 1 second here. All this stuff that's going on right here, that involves the T cells, we call this cell-mediated immunity. Whereas over here, the stuff that are involving the B cells, we call this antibody mediated. Okay, or humoral, I don't know why it's called that, immunity. Okay, so adaptive immunity has 2 branches, the cell-mediated branch that involves T cells and the antibody-mediated, which is involving the B cells. Okay. All right. Let's talk about this stuff. Any questions here? I mean, I think most of them are pretty self-explanatory, but does anybody want me to take up anything? Any requests here? $12 and $17. Okay. I'll do 12 verbally because we kind of talked about it already, right? Describe the role of APC. So this is the APC, right? The role of antigen presenting cells is basically to engulf the pathogen, display it on their surfaces, and then present it to the naive T cell, right? So by doing so, it's able to activate the adaptive immunity. So if you don't have an APC in your system, you would not be able to activate your adaptive immunity. Okay, so that's basically the role of APC. Okay, it's a link between innate immunity and adaptive immunity. Okay, so 17, explain the difference between active and passive immunity. Yeah, this was covered in class, but you know what, let's just do it again because many people have confusion about it. And let's just, you know, make sure we understand it properly. So we have immunity. Okay, and the immunity is divided into innate, which is the stuff that you're born with, and the adaptive, right? Now, under adaptive immunity or acquired immunity, it's separated into active and passive. Okay, so for active immunity, we have I mean the meaning of active immunity is that the host, you are the host, okay? The person who gets infected by whatever, that's the host. The host makes their own antibodies. Okay? In passive immunity, the host receives antibodies from from another source, we would say. Okay, it doesn't have to be another person, it could be like from the animals, I think. Okay, so that's what active is and that's what passive is. Now, because you are making the antibodies yourself, that means you got to go through the whole antigen presentation. You got to go through the activation of T cells and then, you know, the clonal expansion and all that. This is going to be not immediate. Okay, you will not be able to have an effective immune response immediately. But once you have it, it will be long lasting. Okay, permanent is a strong word, but you know, it's pretty long lasting. Some depends what it is. Sometimes it's like lifetime protection. Okay, sometimes it's like good for like 20, 30 years. And sometimes is, you know, maybe it's good for half a year or so, like the COVID vaccine, right? For example, it's not very long-lasting protection in that case. And it could be due to various reasons, okay, which we're not going to go into. Passive means the host received the antibody. So as soon as you get the antibodies, it will have immediate protection, but it will be it would not be long lasting. What's the opposite of long lasting? It will be short term, right? Short term. Yeah, thank you. Sorry. Lost for words for a second there. Yeah, temporary. Thank you. Now we have a whole class of thesaurus here. Excellent. So the active and the passive, they both can be further divided into two additional categories. They could be natural or artificial. Another word for artificial is induced, okay? So natural or induced. So natural is exactly what it means. Is it just happening on its own, right? Through natural means. Induced means is, you know, artificial. Usually it involves some kind of injection, okay? Like you receive a needle or something. Okay, so let's go through the most common ones that people understand or not understand, but like the most common one that people have experience of. Okay, and that's natural active. Natural active would be catching something. Okay, let's say you catching chicken pox. I don't know why I like to use chicken pox as an example. Catching chicken pox, recovering from chicken pox, and now we have monkeypox to worry about, right? But you know, chicken pox, you recover from chicken pox, and then now you are protected for life. Okay. So when I was, my mom told me when I was a baby, less than 1 year old, I caught chicken pox. Okay, whole body covered with dots, not a single spot of good skin. She said, and she had to hold me so that I would because she said putting me on the bed would irritate the pox, and then I would start crying. So she had to hold me and so that I can fall asleep. But eventually I recover, and now I'm protected from chickenpox, right? Okay, so some of you who might be a little bit younger, you might not have caught chickenpox. But how do you get protection from chickenpox? Did any of you get chickenpox? That you know that you caught chickenpox and recover through natural means? Anybody remember? Yeah, okay, some of you good, some, yeah, good, okay. So maybe you guys are not as young as I thought. Very good. Or who knows? Maybe you're young and then you still caught it. Okay, I don't know. You know, you might have caught it for various reasons. Okay, but my daughters, for example, I know for a fact they're not going to catch chickenpox through natural means, because on the immunization recommendation in Ontario, the yellow card, getting vaccinated against chickenpox is a recommendation, and both of them are vaccinated against chickenpox. So that would be an example of induced active immunity. Okay, so that is vaccination against, chickenpox. Okay, against doesn't have to be chickenpox, against COVID. Okay, COVID-19, right? Against whatever, right? Like measles, right? Mumps, right? The MMR, for example, right? The measles, mumps, rubella vaccines, the 3-in-1. All these are examples of induced active immunization. Okay. Now, since we're talking about vaccines, right? Let's do a poll about the vaccine stuff. Okay. Because it's interesting. Oops, I erased the whole thing, and it's no longer that interesting if I erased everything. Give me a moment. I usually like to do this poll, in class with people, but let's do it on in an online environment as well. Okay, because there's a lot of misconception out there about vaccines. You might hear misinformation nowadays. It's good to talk about them. So let's do a poll. Okay, true or false? Okay, A is true, B is false. Okay, A is true, B is false. Vaccine contain live virus or bacteria. Do you think that's true or false? Okay, let's add a C. Let's say sometimes true. Okay, sometimes true. What do you think? You change? Yeah, okay. I wouldn't know which one you picked though, Alex, but yeah, sure. Okay. Anyone else want to pick something? Okay, very good. All right, so it looks like we have a three-way split here. And the answer is, it depends on the vaccine, okay? Some vaccines do contain live virus or bacteria. We call them live attenuated vaccines, okay? So these tend to be the most effective type of vaccine because they are the actual virus or bacteria that is causing the disease. So they're the closest one to the natural version, but they have been attenuated, means they have been weakened, okay, or inactivated so that they no longer going to cause the disease, but they are still alive, and they're the best one to use to educate your immune system. Okay, but because they are live viruses and bacteria, we tend to be more cautious with them. They're not particularly suitable for some people. For example, they would not be suitable for people who are immunocompromised. Okay, they are not suitable for pregnant women. Okay, and so even though they are very good in terms of creating a strong immune response, they are not always going to be the best form. Okay, so some of them contain live viruses and bacteria. Does anybody have any examples of a live attenuated vaccine? Anybody have an example that they know of? Yeah, flu shot. Okay, sometimes flu shots they have the live virus in them. That's very good. Anybody has a big scar here on their arm? Mine is really ugly. It's like super huge. But anybody know what I'm talking about? Like a little small circle or maybe a little bump in the center. A scar on the upper left arm. This would usually be common for people who is raised in Asian country. Do you guys know what that is for? What vaccine gave you that scar? Not smallpox, unless you're like quite old. I don't think you received the smallpox vaccine. That's something that happened, almost a century ago. TB, that's correct. Okay, very good. That is the TB vaccine, the BCG vaccine, actually. Okay, and BCG vaccine is to protect you from tuberculosis. And this is a situation where they actually use the cow version of the bacteria. The TB is caused by bacteria, by the way. And they inoculate you with the cow version, the attenuated cow version of the bacteria, and that in turn gives you protection for the human version. It's not a very good vaccine. The protection is only ranges anywhere from 60 to 80%. And because it's a live vaccine, a bacteria in the vaccine, again, it's not suitable for people who have immunocompromised situation and stuff like that. Okay, but that's what the scar is. Okay, for some reason, like my scar is very big and it's very lumpy. It's funny because when I got the when I went to get my COVID shot, the doctor, I rolled my sleeve, and then before she put the needle, she's like, "What happened here?" And I told her it's from the BCG vaccine. And then she's like, "Usually it's not supposed to look like that." And I'm like, "Yeah, I know it's not supposed to." Well, what did she want me to say? I can't do anything about it. It's not like I can cut it off or anything like that. Anyways, I'm not going to show you the scar because it might traumatize you a little bit. Anyways, next one. Flu shot. Flu shot can give you the flu. What do you think? Can flu shot give you the flu? My mom always says, I don't want to get the flu shot because I always get sick from it afterwards. I always get the flu. Is that true or false? Or maybe sometimes you can. Yes, A is true, B is false, and C is sometimes. All right. Ooh, we have 14 responses. Very good. Okay, this is a hard false. Okay, 100% false. Okay, you cannot get sick. You cannot get the disease from the vaccine that you are trying to get the protection for. So if you're getting the flu shot, you're not going to get the flu. If you're getting the COVID vaccine, you are not going to catch COVID from the COVID vaccine. Okay. However, sometimes, well, yeah, you get side effects, but side effects is not the same as catching the actual diseases, right? Okay. You do get the fever, you do get the eggs, but that's just because you're introducing foreign substances into your body, right? Okay. The fever and the egg, like the body eggs, they are not caused by the actual disease, right? They are just the side effects of the vaccines. Okay. Now, some people say, okay, I got the flu shot and then I actually got the flu the next day. Okay, or like I got the COVID shot and then I had COVID 2 or 3 days after. The vaccines themselves, they do not work immediately, right? As you just learned from this class, it takes time to make the virus. It takes time to make the antibodies. You got to select the T cells and so on and so forth. All that takes about a week or so, right? So the immunity is not immediate. which means during that time, if you actually come in contact with someone who's infected with the flu or with the COVID, you are still susceptible of catching the disease. So if you get the flu shot and then you have the flu the day, 1 or 2 days afterwards, you got the flu not because of the flu shot, but because of your exposure to someone who actually has the disease, okay? So it's absolutely 100% impossible for you to catch any of the diseases from any of the vaccines that are out there, okay? Now, if you do a search on the internet, you will find that, there are a few unfortunate incidents in the past where, for example, the polio vaccine, which is live attenuated, there was a little bit of manufacturing error that the virus was actually not inactivated in the vaccines, and it was given to patients, and that inadvertently actually gave people polio as a result. But that was because of a pharmaceutical error, right, by the company, not because of the fact that vaccines can give you the disease. All right. And one last one here. Mercury is used as a preservative in vaccines. What do you think? True or false? This is the one that the anti-vaxxers are saying, well, there are mercuries in the vaccines, and it's poisonous. You're injecting poison into the body. That's one of the key arguments from them. Is it true? Or is it not true? Or sometimes true? Okay, I'm just going to wait a little bit for more people to answer because you guys have been quite responsive here compared to the other poll questions. Okay. So who chose sometimes? Can you give me an example of when do you think is used and when is it not? If you would like to. Okay, go ahead.
I know it's used in all vaccines.
Sorry. I think you're cutting off a little bit. Would you mind repeating yourself, please?
Sure. To my knowledge, it's still used in some vaccines in very small amounts.
Okay, yeah.
Not childhood illness vaccines, though.
Okay. Sorry, Tyler. I think your mic might not be working.
Okay.
All right. I think I caught a little bit of what you said though. So yeah, sometimes it is actually used in for example a repeat vial. Okay. So like the flu shot, for example. Okay. So they have a vial where you would draw the vaccine from. Okay, using needle, you would draw like 10, 20 times from it and give it to 10, 20 people. And because you're repeatedly drawing it from the same vial, it's important to include some preservatives, right? So that you don't get contamination, right? And so the preservative that is being used is what we call the thimerosal in some cases, and the thimerosal does contain the mercury atom. Okay. But it's very different though, okay? Like having an atom in a molecule does not mean it's going to have the same property as the atom, right? As you learn in chemistry class, table salt is sodium chloride. Sodium is super explosive, chloride is super poisonous, but when you put them together, it's like table salt, right? So, just because it contains a mercury atom does not automatically mean the whole thing is going to be poisonous, right? Okay. And a very important distinction to understand is this type of molecule, it is insoluble in fat, okay? Which means once it goes into your system, it will pass out really quickly, okay? In the urine, for example, okay? So this type of mercury is different from the kind of mercury that you will find in your in like a seafood. Okay, they say you don't eat too much tuna or saltfish or something like that because they're very high in mercury, right? So those kind of mercuries, they are fat soluble. Those are the methyl mercury that we talk about, and they do accumulate in the body and cause harm, right? But these are ethyl mercury, which they will not stay in the body. So even if you get some of these in your system from the vaccine, it's just going to come out really quickly and will not cause any harm. All right. But you know what? Don't believe everything I said, right? Be a critical person. Go do your own research and make sure the source of the research that you are doing is a credible source and not some, blog or some websites that are not trustworthy. In any case, let's go back to here. I went on a huge tangent. Sorry about that. Next, we have the passive, right? So what's natural passive? Natural passive is like, for example, from the mother. Okay, so mother to child, either through placenta. We know antibody can pass through the placenta, or breastfeeding, exactly. Or breastfeeding. Okay, so if the mother gets a COVID vaccine, she has immunity, right? She could pass it through to the baby, and they could continue to pass on that immunity through breastfeeding. There is an article that came out, I think I read it last month, that says, mothers who are vaccinated, their babies are also I forgot what the exact percentage is, but they are significantly more protected from COVID than mothers who are not vaccinated because of this ability for the mother to pass it on. Okay. Induced. What is induced passive? Induced passive is a situation where I need to have protection. Well, okay, so first of all, let's just clarify. For induced passive, we can use it for prevention or treatment. Okay, so prevention is like this. I need to go to certain places, okay, and like out of the country, and in that country there is going to be a very prevalent disease that I don't have protection against. But because I'm going on a business trip, I have to go like tomorrow, so I need to have the protection right away. So what I would do is I would receive a serum. Okay, so the serum would contain antibodies that would protect you from that disease. Okay, and as soon as I receive it, I will be protected, and then I go on my trip. I hopefully won't get sick. Okay, but after I come back, the antibodies will eventually break down, and I will lose that protection. Okay, so I have immediate protection when I receive the serum, but it's short-term protection. Okay, so another one is treatment. Last October, I was out for a run in my neighborhood, and then a dog bit me. Okay, so it was someone was walking two dogs actually, and I don't know for what reason. I didn't even look at the dog. Okay, like I ran by the dog, and then it just lunged at me and then bit me right in the abdomen. And so I don't know why I did not stop the person and ask questions. I just told her your dog bit me, and then I kept on running, and then afterwards I see there was a bite wound and it was bleeding. So I went to my doctor, and the doctor said you have to get the rabies serum. Okay, because you don't know if the dog has rabies. The thing about rabies is if you actually catch rabies and you don't treat it quick enough, you are almost guaranteed to die from it. Yeah, exactly. You will die. And as you can see, I'm very much alive, so I did not catch the rabies. And what happens is the doctor immediately gave me a serum. Okay, so he ordered it from public health, and then they immediately have someone taxi the thing to the doctor's office, and then I was called in, and then I received actually 4 shots on that day. Okay, and that's basically serum that contains anti-rabies antibodies. Okay, so in case the rabies virus is in my body, that serum will prevent the disease from manifesting itself. Okay, I also received a shot for tetanus as well. Apparently, the bacteria that causes tetanus can live in the dog's saliva. So if the dog bites you, you could get tetanus that way. And my last tetanus shot was more than 20 years ago. So, you know, my immunity may be waning a little bit. So I got that as well. And then for the entire month, I have to go back every week to receive an additional rabies shot. So in total, I received 4, 8, 9 shots, okay, thanks to that dog. And now every time I run and I see the dog, you know, coming anywhere near me, I rather run on the street. And then my wife jokes and say, "What, you get run over by a car next time instead?" So, you know, it's a scary world out there. That sounds like a scary experiment. Yeah, yeah. Well, you know, I really don't know what I was running through my mind at the time. But had I stopped and talked to the dog person, and exchange information, then maybe I don't have to receive the, you know, the shots, because all dogs are supposed to be vaccinated, right? As per Ontario law. But you know, you never know. Okay, you know, even if I exchange information, maybe the person lied, right? Then I end up dying with rabies. That would be terrible. Okay. Anyways, that would be an example of treatment, right? So many serums exist for treatments like these. Hepatitis. Okay. Let's say you accidentally get jabbed by a needle that you pulled out from someone who has hepatitis. You can get a serum. Okay. Dogs are not the only animals that can spread rabies. Do you guys know any other things that can spread rabies? Other things that is you know, in the neighborhood, like, yes, raccoons. Okay, those pesky little things. If you get scratched by a raccoon, or worse, get bitten by it, you should also do that. Birds, I'm not so sure that if they could spread rabies. Maybe. I have to double check that one. But bats is another one. Okay, bats. I remember when I was still in university, and you know, me and my wife, we were still dating at the time. She called me one night. It was 2 a.m. in the morning. Okay, and then she called me. She's like, "Oh, you gotta come help me. There is a bat in the house." And then I'm like, "What are you calling me for? There's nothing I could do, right? Like, you know, call animal control instead." But it was 2 a.m. in the morning. I didn't end up going. I just you know, hung up. She was mad at me for some time, but eventually she caught it. Okay, she caught it with the garbage can. Okay, she said somehow the thing was perching on the window or something, and she just like used the garbage can to catch it, and was able to dispose of it. But then, you know, all jokes aside, afterwards we're saying, well, we don't know how long the bat has been, you know, living in the house, and you know, it's possible that you could catch the rabies from them. So, you know, lots of things to watch out about. Yes, skunks. Yeah, all those things could potentially spread deadly diseases. Anyways, I went on a lot of random tangents here, but hopefully that answers your question, whoever asked that. Okay, let's go to the application questions here. All right, here, Joffrey. Joffrey is bitten by a rattlesnake while on a camping trip. His friends immediately apply ice packs to the bitten area to slow the spread of the protein-based toxin, then rush him to an emergency facility. What treatment would provide immediate protection, and why? What do you think? What are the people gonna give him? Yes, they're going to give him a serum. Okay, they're going to give them an anti-venom serum. You know, they say if you get bitten by a snake, you have to remember what the snake looked like. Okay, best case scenario, you will take a picture of the snake, right? Okay, or you have to be able to describe the snake, because depending on what kind of venom it is, you're gonna have to apply different kind of serum, right? There is no universal serum for all the venomous snake out there, right? You have to you know, find the right serum for that, and they have to know what kind of snake bit you before they could give you the right serum. Is it counterintuitive to put ice pack on the snake wound? That will make it worse. I don't know why do you say that? Is there something that you You read that, you know, the ice would make it worse. I'm not aware of that. Okay, I'll wait for Alex to respond while we move on to the next part. Explain the type of immunity this would be. Well, because it is antibodies produced by someone else and it is not natural when you inject something, this is going to be induced passive. Okay. If Joffrey were to be bitten by another rattlesnake in the future, unlucky Joffrey, will he be immune to the toxin? What do you think? Yes or no? Nope, that's right. Because this is passive, and so what's going to happen is the immunity is going to be weighing afterwards, right? The antibodies will be gone, and then he will need another serum. I actually asked the doctor the same question, even though I know the answer to it. But you know, sometimes, you know, asking a doctor is more reassuring. I was like, if I get bitten by another dog, am I gonna have to come in and get all these shots again? And he says, yes, unfortunately, you would have to. So that's why I'm gonna stay away from dogs. Okay, Alex says ice or cold would make the capillaries to contract and contract is best. Oh, well, if the cold actually would make the capillaries contract, you're correct. But contracting will stop the blood flow, right? It would basically not allow it to spread further, right? And Tyler said ice causes smaller blood vessels to constrict, which can apparently exacerbate tissue damage caused by the venom. Oh, okay, so there you have it. Well, in the general area of the bite, yeah. Okay, I did not know that. Okay, so his friends were apparently not very knowledgeable about you know, snake bites. But thanks for the information. I will definitely look that up and see what's the proper first aid treatment if you do get bitten by a snake. We don't have a lot of snakes here though, so you know, not to worry too much. Next one dose of vaccines against chickenpox will virtually offer a lifetime protection against the virus. Now, I do want to say some people get shingles, right, which is caused by chickenpox virus as well, but that's another story. Whereas a new flu shot is needed every year. Explain the reason for this. Does anybody know why we need to have a flu shot every year? Mutation, that's correct, okay? So for those of you who do not know, there is our flu virus, our influenza virus. And on the surface of the influenza, we actually have two very distinct antigens. We have the H antigens, the hemagglutinin, that's what it stands for, and then the neuraminidase, the N, okay? There are 18 possible versions of the H and 11 possible versions of the N. So if you do the math, right, 18 times 11, cannot do mental math right now. Let me plug that into the calculator. That is 198 different possible combinations for the flu virus. Okay, so each year, the WHO, they predict the dominant strain that is circulating in they will predict the dominant three strains that will be circulating in the upcoming season. So for example, they will predict, okay, strain A, B, and C are the dominant strain. And then they will prepare the vaccines, or the pharmaceutical company will prepare the vaccines for those three strains. And sometimes they are wrong, okay? If the strains that are dominating would be like D, F, and K, whatever, right? Then the vaccines would have very, very low efficacy. Okay, so we are a little bit more fortunate living in the northern hemisphere because people in the southern hemisphere, Australia, for example, they would have their winter before us. So we can take a look at how protective the vaccines are in their winter, and then that would give us a sense of how much protection we will get for our upcoming winter. Okay, so that's why you would need a new one every year. So anybody know any flu strain that are out there? Like, again, you might read them on the news paper or hear them on the radio, with the H and the N. Usually is a number, right? H something, N something. What are some of the common ones that you guys know of? Just want to see if anyone is aware of these things. yeah, Rureda said H1N1, right? That's the swine flu, right? They call it, but actually it's the Spanish flu that happened in 1918, right? But then there's a recombinant version that resurfaced recently, right? That is the swine flu. There's another one that's common one. Anybody know the bird flu one? The bird flu, the avian flu. What number was that one? If I say it, you probably will be like, oh yeah, I remember hearing about it. Right, the H5N1, anybody heard of that before? Maybe, maybe not, but that's okay, right? Well, now you know what the H and the N stands for, okay? The H and the N right here. There is a drug called Tamiflu. I don't know if you've heard of that. Tamiflu will block one of these surface markers, causing the flu virus not be able to infect your cells, and therefore it will render the flu harmless. Okay, moving on. HIV infects macrophage and destroy them. Explain how this will affect your immune system. Well, as you know, the macrophage is actually an APC, right? So if you destroy the APC, you will not be able to mount an effective adaptive immunity because you cannot activate the naive T cells. So you eventually only have innate immunity. So people with HIV, people with AIDS, they die from simple infections that normal people or healthy people, I should say, healthy people would be able to fight off easily. So things like common cold, things like perhaps pneumonia, right? All those things, they're not able to mount an effective immune response against them because they lack the macrophage. Okay, some of the COVID vaccines that are being developed. Okay, so at the time when I make this, they are still being developed, but now obviously they're all very common. They are mRNA vaccines based on your understanding of cell biology and immune system. Speculate how mRNA vaccine works. Okay, so there is another type of cells that is also APC. I did not teach you yet. These are called dendritic cells. Okay, and they look like little star-shaped cells. So what happens is you will inject the mRNA. Okay, so this is the mRNA, and then the mRNA is going to go into these dendritic cells. The mRNA, if you read anything about this, they're actually surrounded by little lipid pockets, bubbles, and then they will go into the dendritic cells, and then the dendritic cells will translate the mRNA, and they will make all these spike proteins, the ones that are found on the COVID virus, okay, on the surface. Okay, and then what they will do is they will present it to the naive T cell, right? So then you will make a bunch of antibodies that will recognize the spike proteins. And when the actual COVID virus comes in, the SARS-CoV virus comes in, the antibodies is going to be able to recognize them and hopefully neutralize them before they cause too much damage. Okay. Again, the protection is good, but it's not 100%, right? And you might still show some mild symptoms even after vaccination. In fact, I know a few friends and actually coworkers who still got the COVID after getting 2 doses or 3 doses of vaccines, and they still have some symptoms like sore throats, fever, and that kind of stuff. But they do recover, relatively quickly. Okay, so that is that. Any additional questions? I still have some polls here if you guys want to do them. And I hear you say you do want to do them. So let's go through them over here. No Kahoot today. Okay, maybe next time. Maybe next time. Number 6. This is select all correct statement. Okay. So let's go through them. Let's see. Antibodies are Y-shaped proteins. That's correct. Antibodies produced against one pathogen will generally work against another pathogen. That's not true. Okay. They usually would only protect one type of a pathogen. If you get antibodies that work against hepatitis, it will not work against I don't know, Ebola or something. However, sometimes there are some crossover, right? If the viruses are related enough, they will work against each other. For example, the monkeypox is a cousin of the smallpox. So they are actually rolling out the smallpox vaccine in the UK to fight this monkeypox outbreak. And so in that case, because the two viruses, even though they're different, they're related, they're similar enough that when you make antibody for one, you're gonna have a protection for the other one. Antibodies are produced by plasma cells. We know that is true. Antibodies are found in the blood plasma, also true. And variation in antibodies arise from gene shuffling. That is also true. Okay, so except for B, everything else is true. Okay, let's try another one. Sorry, some hospital, some. That would be artificial passive, right? Okay, because you are receiving antibodies from someone else and you have to be injected with them. So it's artificial passive. Of course, they no longer do this, right? But in fact, this is something that they would try for very deadly diseases when they still don't have a vaccine, like Ebola. That's something that they did during the Ebola outbreak in 2018, I think, right? Or was it 2017 or 2018? I don't remember, the one that started in South Africa. Okay, this one, why do we need to receive a flu shot each year in order to have protection against the flu virus? We talked about that already, right? Each year, the dominant strain is different. Okay, last one. This one is particularly lengthy and difficult. All right, I'm gonna stop it right there. So this is something that they actually are experimenting with, and some type of cancer, they actually are at the clinical trial stage stage for something like that. Okay, so they take the T cells out of the body, they kind of educate them about what the cancer cell looks like, and then they put it back into the patient, right? And the idea is that now they know what the cancer cells look like, they would go and attack them. So for this to work, the receptor must do all of the following except, number one, recognize an antigen that is found only on cancer cells and not other somatic or bodily cells. So that's something that it needs to do. We don't want it to indiscriminately attack all the cells. We only want them to attack the cancer cell. Next one, recognize an antigen that constantly mutates in the cancer cells. That would be the answer. That's the exception. It's not possible for a single receptor to keep on recognizing something that keeps on changing, right? Okay, there's a key and lock relationship between the antibody and what it recognizes. If its target is mutated, it changes, then the antibody that you produce or the receptors that you have will no longer work, right? So, you know, for something like this to happen, it's not possible. That's why it's the exception. Trigger clonal expansion once it binds to the target antigen. That's true. We want to have those huge colony of T cells, where they would then differentiate into the helper T cell, killer T cell, so on and so forth. And we definitely want them to become the effector cells that will attack the cancer cells. So the answer is this one. Okay, that's it for this week. The fill in the blanks right here. Again, I will just paste the answer from before, and then you guys can check it yourself. I will only teach you guys one more lecture, right? I'll teach you lecture 4 next week. And then we will have a review on the following week, and then the test will be on that week as well. And then Muren will come and take over the course. Okay, so just hang in a little bit longer, hang in there. We only have one more lesson before the first test. Okay, thanks again for your attention. I'll see you all next Thursday. Okay, bye for now.